Bismillahirrahmanirrahim. Assalamu alaikum. Today I welcome all my students of second year MBBS and BDS to the topic that is pancreatic secretions. Now these secretions they are very important because they have one of the major roles during the course of digestion of food. Now, what is pancreas? You all know that pancreas is actually a leaf like structure which is lying parallel to and beneath the stomach in a large compound gland with most of its internal structure which is very much like that of the salivary glands. The pancreatic digestive enzymes they are being secreted by pancreatic acini in large volumes of sodium bicarbonate solution. They are being secreted by the small ductules and larger ducts leading from the acini. The combined product of enzymes and sodium bicarbonate then flows through the long pancreatic duct, which is normally joining with that of the hepatic duct immediately before it empties into the duodenum through the papilla of water, surrounded by the sphincter of Oddi. See the leaf like structure, the major pancreatic duct, the accessory pancreatic duct, then there is a common bile duct. See this leaf like structure lying horizontal parallel to that of the stomach below it and secreting lot of enzymes along with that of the sodium bicarbonate in the secretions. The ampulla of water, also known as the hepatopancreatic ampulla, is formed by the union of the pancreatic duct and the common bile duct. Pancreatic juice is secreted most abundantly in response to the presence of chyme in the upper portions of the small intestine. The pancreas is secreting insulin as well, but this is not secreted by the same pancreatic tissue which is secreting the intestinal pancreatic juice. Insulin is secreted directly into the blood, not into the intestine, by the isles of Langerhans that occur in isle patches throughout the pancreas. Pancreatic digestive enzymes. Pancreatic secretion is containing multiple enzymes for digesting all the three major types of foods, which are proteins, fats, carbohydrates. And at the same time, it is containing large amount of bicarbonate ions, which have the role to neutralize the acidic um, composition of the chyme which is being delivered or emptied from the stomach into the duodenum. So it is going to neutralize it. The most important of the pancreatic enzymes for digesting proteins are trypsin, chymotrypsin, carboxypolypeptidase. By far the most abundant of these is trypsin. Trypsin and chymotrypsin split the whole in partially digested proteins into peptides of various sizes but do not cause release of individual amino acids. Carboxypolypeptidase splits some peptides into the individual amino acids, thus completing digestion of some proteins all the way to the amino acid state. The pancreatic enzymes for digesting carbohydrates is pancreatic amylase, which hydrolyzes starches, glycogen, and most other carbohydrates except cellulose to form mostly disaccharides and a few trisaccharides. The main enzymes for the fat digestion they are the pancreatic lipase, cholesterol esterase, phospholipase. Pancreatic lipase is capable of hydrolyzing neutral fat into fatty acids and monoglycerides. Cholesterol esterase is causing hydrolysis of cholesterol esters. Phospholipase is splitting the fatty acids from the phospholipids. The first synthesized When first synthesized in the pancreatic cells, the proteolytic digestive enzymes are in the inactive forms, which is trypsinogen, chymotrypsinogen, procarboxypolypeptidase, which are all inactive enzymatically. They become activated only after they are secreted into the intestinal tract. Trypsinogen is activated by an enzyme called enterokinase, which is secreted by the intestinal mucosa. When chyme comes in contact with the mucosa, also trypsinogen can be autocatalytically activated by the trypsin that has already been formed from previously secreted trypsinogen. Chymotrypsinogen is activated by trypsin to form chymotrypsin, and procarboxypolypeptidase is activated in a similar manner. See, what happened is that the procarboxypeptidase is activated to carboxypeptidase, which is then activated to chymotrypsinogen is going to change to chymotrypsin, then trypsinogen to trypsin, and that is actually by membrane bound enterokinase enzyme, which is being released from the epithelial cells. So, all these inactivated enzymes they are being activated in the presence of enterokinase. Secretion of the trypsin inhibitor prevents digestion of the pancreas itself. It is important that proteolytic enzymes of the pancreatic juice not become activated until after they have been secreted into the intestine, because the trypsin and the other enzymes would digest the pancreas itself. The same cells that secrete proteolytic enzymes into the acini of pancreas secrete simultaneously another substance called trypsin inhibitor. This substance is formed in the cytoplasm of the glandular cells, and it prevents activation of trypsin both inside the secretory cells and in the acini and ducts of the pancreas. And because it is trypsin that activates the other pancreatic proteolytic enzymes, trypsin inhibitor prevents activation of others as well. When the pancreas becomes severely damaged or when a duct becomes blocked, the large quantities of pancreatic secretion sometimes become pooled in the areas of pancreas itself and is going to do the damage to the pancreatic tissue itself. And under these conditions, the effect of trypsin inhibitor is often overwhelmed, in which case the pancreatic secretions rapidly become activated and can literally digest the entire pancreas within few hours times, giving rise to the condition called acute pancreatitis. This is sometimes lethal because of accompanying circulatory shock. Even if not lethal, it may lead to a permanent lifetime pancreatic insufficiency. Secretion of bicarbonate ions. The enzymes of pancreatic juice are secreted entirely by the acini of pancreatic glands. The other two important components of pancreatic juice, bicarbonate ions and water, are secreted mainly by the epithelial cells of the ductules and ducts which lead from the acini. When the pancreas is stimulated to secrete copious quantities of pancreatic juice, the bicarbonate ion concentration can rise to as high as 145 milliequivalents per liter. This provides a large quantity of alkali in the pancreatic juice that serves to neutralize the hydrochloric acid emptied into the duodenum from the stomach. See how the formation of bicarbonate occurs and how it is being delivered by the process into the lumen. The basic steps in the cellular mechanism for secreting sodium bicarbonate solution into the pancreatic ductules is this: that what happens is that the carbon dioxide diffuses to the interior of the cells from the blood, and under the influence of carbonic anhydrase is going to combine with that of the water and form a carbonic acid. The carbonic acid is in turn going to dissociate into bicarbonate ions and hydrogen ions, and then bicarbonate ions are actively transported in association with sodium ions through the luminal border of the cell into the lumen of the duct. The overall movement of sodium and bicarbonate ions from the blood into the duct lumen creates an osmotic pressure gradient and is going to cause the osmosis of water also into the pancreatic duct, thus forming an almost completely isoosmotic bicarbonate solution. So this is how the release of bicarbonate ion taking place. Then we have the regulation of pancreatic secretion, that is um, going to be dealt with inshallah tomorrow. Until then, stay tuned, give a good reading to the topic, and inshallah tomorrow we'll complete the topic of pancreatic secretion. So till then, stay blessed and Allah Hafiz.